I chose John Abelson as my role model, and John completed his undergraduate degree at the Washington State University, where he received a Bachelor's of Science in Physics in 1960. He then later went on to graduate from Johns Hopkins University, where he received his PhD in biophysics in 1965. After that, he completed his postdoc fellowship at the MCR Laboratory of Molecular Biology in Cambridge, where he actually studied under Francis Crick, and together they worked on nonsense suppressors in E. coli. So the two things that I admire most about John would be his tenacious drive and his work ethic, as well as his creativity. His tenacious drive allowed him to study at some of the most prestigious schools in the world, and it also allowed him to study under some of the greatest scientific minds to date. I also admire John's creativity, as I previously said. And before the function of the splicesome was completely understood, he studied the structure of it in order to deduce its function, which I think is an intuitive, inherently scientific way of thinking. So I really admire how he was able to work around not knowing what something is in order to decipher such a complex function solely by studying the physical structure. So overall, his creativity is something that I really strive to emulate in my own work and my own future work. So John has studied and worked in a few different fields of science. He primarily does his work in molecular biology, but he's also worked in the field of biophysics and biochemistry. So John has published a whole host of papers, and his most recent paper was published in 2001, and the title is The Crystal Structure of a Dead Box Protein from the Hyperthermophile Methanococcus jannaschii. So what that is they found a dead box helicase within this organism. And as we studied in class, the helicases are the enzymes that kind of unzip DNA during DNA replication. So the major things that John has been able to discover thus far was that he was an integral member in determining the mechanism for RNA splicing. So he actually, his studies showed how a transcript may be made into an mRNA or a tRNA molecule through the presence of certain introns, and then he kind of helped shed light on how those fragments of DNA, the exons, are spliced together in order to form a functioning nRNA molecule, which would then lead to the formation of a protein. So he also, as I previously mentioned, identified and discovered the structure of the splicesome, and then he was able to deduce that overall function of it. So as you can kind of tell, most of his work has to do with transcription, and like transcription within various organisms, and he primarily worked with yeast. So that was just a quick little summary about John Abelson, his past, and some of his major studies that have influenced the scientific community. So once again, I really admire him because I think that he has a brilliant mind, and I think that he's very creative in the way that he figures out the unknown and tries to answer his questions, and that's something I really want to try to imitate in my future work, hopefully, and so I can possibly try to discover something somewhat as significant as he did.